So continuing here with part 2 of the microbial growth lecture. The terms on this slide are very important. You need to know every single one of them. These are the terms that we're using for the oxygen requirements of different microbes. And so here are the terms. Obligate aerobes, those are organisms, they must have oxygen. They will not grow if there's no oxygen or not enough oxygen available. Then we have facultative anaerobes. They are best adapted to just about any environment. They can grow with or without oxygen. They can switch their metabolism. They can grow via fermentation, or they can also do anaerobic respiration with no oxygen. So they have a lot of options. Obligate anaerobes, they are to them, oxygen is toxic, and so they have to grow in conditions where there is no oxygen, because oxygen is literally toxic to them. And we'll take a look at how that works. We have aerotolerant anaerobes. They can tolerate some oxygen, but they can't use oxygen for their growth, but they're not necessarily killed by oxygen either. The microaerophiles, they require a little bit of oxygen, but not as much as you would find in the air, and so they'll kind of hide out in an environment where there's a little bit of oxygen, but not that much. So here are the different oxygen requirements that you find in bacteria. Let's start with the obligate aerobes right here. They must have oxygen. They can only grow in the presence of oxygen. Those are the ones when you grow the bacteria, they will be hiding only on the top. They don't even grow anywhere on the bottom here. You will see no growth right here. And if you remember, we were growing Pseudomonas aeruginosa in the lab. That's an obligate aerobic bacterium, and that one will only grow on top. And so even if you do a motility stab, you won't find any growth, even though it has motility, but you won't find growth because this bacterium does not like to grow when there is not enough oxygen down here. Then here we are the facultative anaerobes. These guys grow everywhere. They tend to grow better when with the presence of oxygen, so they can do aerobic metabolism, which is usually more efficient in generating energy, and so they like the oxygen, so you'll find a lot of them on top, but you will find growth throughout the entire test tube. Here's the opposite extreme to the obligate aerobes. These were the obligate anaerobes, and these guys only grow here on the bottom. They really hate oxygen. In fact, they're harmed by oxygen. Then we have the aerotolerant anaerobes. They really grow anywhere. They can tolerate oxygen, but they are classified as anaerobes, but they don't even have a preference to grow up here. So that's the difference to the facultative anaerobes. They just really will occupy the entire test tube right here. And then we have microaerophiles. They like a little bit of oxygen, but they don't want as much as we would have in the atmosphere, so they will settle for growth somewhere in the middle of a test tube right here, where it's a little bit of oxygen, but not that much. So let's take a look at some toxic forms of oxygen. These toxic forms of oxygen are the reason why some bacteria are obligate anaerobes, and they don't have enzymes that will get rid of these toxic forms of oxygen like other bacteria do, and so that's why these they're highly reactive forms of oxygen, they will harm cellular structures. For example, the singlet oxygen, this one is a highly reactive form of oxygen. Then, very important, the superoxide radicals. These O2 minus, and then here the dot denotes that it's a radical. And here we have, so this one forms peroxide, actually, an oxygen. Then here the peroxide anion. This one reacts to peroxides forming water and oxygen. So the best way to get rid of these peroxides would be to use an enzyme that just forms oxygen and water. And there is an enzyme that those bacteria that can grow with oxygen, they usually have it. And you should know this enzyme. It's called catalase. So this particular reaction here is catalyzed by the enzyme catalase. And if a bacterium is catalase negative, meaning it doesn't have catalase, then the peroxide will destroy cellular structures. Down here on the bottom, another highly reactive substance is called a hydroxyl radical. Again, the dot here denotes it's a radical. Now continuing here with organic growth factors. So those would be organic compounds that the organism must obtain from the environment that it's living in because it cannot make it on its own. And these would be including vitamins, amino acids, purines that form the purines and pyrimidines. Of course, those are the base molecules for forming nucleotides that then get incorporated into DNA, RNA, and other nucleotide containing organic molecules. Oops. So here we can quickly check our understanding. If bacterial cells were given a sulfur source containing radioactive sulfur S35, where would you find that, and what part of the cell might contain that? What should come to mind would be proteins that contain amino acids that sometimes contain sulfur. How could you determine whether a microbe is a strict anaerobe? Well, you could grow it in different environments and see where it grows. And oxygen is so pervasive, how could a organism avoid any kind of contact? Well, it's going to have to grow at a place where there is no oxygen, because in this case the oxygen is simply toxic to the organism, because usually it doesn't have enzymes that will get rid of toxic forms of oxygen. Okay, so moving on, a little bit about biofilms right here. We talked about biofilms before because there's such a problem in hospitals a lot of times. So biofilms are microbial communities, and it's basically layers upon layers of microbes that are forming where the outer layers of the community shield the ones that are underneath from any kind of environmental damage, and basically protecting the lower layers from chemicals, even heat and other things. And that can be a real problem for instruments in hospitals that have to be that are not single use. So cleaning up instruments that are being used between patients, of course they're trying to autoclave and sterilize the instruments, but if there are biofilms that have formed, it can be a real problem. And so typically these biofilms, they stick to surfaces, they hide out in crevices of instruments, for example, and then the big thing is that they shield the bacteria from environmental factors such as disinfectants, even autoclaving. So here's an example of a biofilm, clumps of bacteria adhering to a surface right here. And again, this can be a real problem if you're trying to disinfect an instrument that has a lot of little crevices. Okay. So biofilms are commonly found in the digestive system and sewage treatment systems. They can clog pipes. They are said to be 1,000 times more resistant to microbiocides than regular bacteria would, because they're shielding the lower layer so well. And here's another bad statistics. They're involved in 70% of infections that are caused by catheters, heart valve transplants, contact lenses, dental caries. So that's a pretty high number. So let's now move on to culture media. Obviously, if we want to grow bacteria, let's say in the lab, then we want to prepare a medium that allows these bacteria to grow. So we want to make sure that we provide all the nutrients that the microbe needs in order for it to grow. And so culture medium contains the nutrients and is specifically prepared for microbial growth. We want to make sure that when we're starting out growing bacteria, that there are no living microbes in there. So we will autoclave these culture medium first, and then make sure that it's sterile to begin with. So here you would have to know the term sterile. Sterile means no living microbes, no life in general. An inoculum means that you're introducing microbes into your growth medium using like a loop or a needle. There's different ways you can do that. And a culture, by definition, means that these are microbes growing in or on a culture medium. We have had in the lab, we had broth, so liquid culture medium. We also have had agars. Those are solidified culture medium. So here, our most popular culture medium in the microbiology lab is agar. It's a complex polysaccharide, and it is not metabolized by bacteria. So they don't like to eat it up like they would gelatin, for example, they would eat it up. But agar, they don't really like it. And so therefore it makes a really good solidifying agent for culture media. And so we call a Petri dish that contains agar, we call it now a Petri plate. We also can put the agar containing medium into test tubes. If we let it harden at an angle, then we call it a slant. And if we just let it harden right side up, then it forms a deep. Those terms you hopefully remember from lab. The important thing about agar, again, is that it is a chemical that is not metabolized by microbes, and so therefore your medium will stay solid. Agar liquefies at 95 degrees Celsius, which is near boiling temperature, and it solidifies around 40 degrees Celsius. Now here are another two terms that you need to know, the difference between chemically defined media and complex media. Let's start out with the complex media because that's the one that you've worked with in the lab before. So in the complex media, we have nutrient agar, nutrient broth, and we're using, to prepare those complex media, we're using usually yeast extract, as an example, extracts of meats or plants. And so there you can expect that there's a variety of nutrition in those in a yeast extract, but it's not chemically defined. So that might be slight variations from batch to batch of yeast extract or some meat extract. Now, going up here to the chemically defined media, those are media where you have an exact chemical composition and you can make it exactly the same way every time. For example, if you have fastidious organisms, remember those are the picky eaters, they might require special growth factors or some special chemicals, and then you need to make sure that you provide them in your medium, in your growth medium. So in this table here, you see a recipe for a chemically defined medium. You can see that all the amounts of each ingredient are exactly spelled out, and then you make that up to 1 liter, a fixed amount, and you can prepare this medium the same way every time. Here's another example. This one is a little bit more elaborate recipe, but also just a recipe for a chemically defined medium for a particular bacterium that needs, it's a picky eater that requires certain amino acids and other chemicals that need to be added, vitamins, trace elements. So again, an example, this is an example of a chemically defined culture medium. And here you have a complex medium, a recipe. This one here will be peptone, partially digested protein, beef extract, so that's a meat extract. This one could vary from batch to batch. Also the peptone tends to vary. And then you have some salt and then agar and then the water. But it will be impossible to make it exactly the same way because the beef extract, depending on the beef, the exact composition there, it will be varying from time to time. Here's an example for culture media required for anaerobic growth. This is a real challenge because oxygen is everywhere, and if you want to grow anaerobes, you need to make sure that there isn't any oxygen in your growth medium. And so typically you would have to add chemicals such as the sodium thioglycolate. That was a very popular one that essentially absorbs oxygen. A lot of times you also need heat to deplete the oxygen. Here is what we were going to do in a lab. And I'll talk more about the anaerobic jar. That will start our next part with the anaerobic jar.